Today's topic is southern hybridization or southern blotting. So, it is a molecular technique where we detect specific DNA sequences within a complex mixture of DNA fragments. It is based on DNA hybridization. The first DNA is what we are detecting, and the another DNA is our probe. It was developed by E.M. Southern. That's why called as southern blotting. So its principle relies on the ability of complementary single-stranded DNA sequence to hybridize. DNA fragments are separated by electrophoresis, transferred to a membrane, that is blotting membrane, and detected using a labeled complementary DNA probe. So first of all, genomic DNA will be isolated. It is fragmented using restriction endonuclease or by a mechanical shearing to obtain DNA fragments. Then gel electrophoresis will be performed to separate the DNA on the basis of its size. Then alkali treatment will be given so that DNA gets single stranded. After that the blotting is performed, in which uh, in a tray, first filter papers are kept, above that gel is kept, and above that blotting membrane is kept, and above that filter papers and the weight is kept. In the tray there is buffer. With the help of the capillary action, the buffer will move upward, and along with the buffer, the DNA, single stranded DNA, will also move upward on the nitrocellulose membrane. After that, baking is performed either by using uh, high temperature or by UV light. In case of nitrocellulose membrane, in case of nitrocellulose membrane, high temperature is used, while in case of nylon membrane, UV based cross linking is performed. After this, the hybridization is performed by using single-stranded DNA or oligodeoxynucleotides, and then washing will be done, and then detection will be performed. So coming to the first step, genomic DNA is isolated and fragmented either by mechanical shearing or by restriction endonuclease digestion to generate DNA fragments of varying sizes. DNA fragments are separated based on size using agarose or polyacrylamide gel electrophoresis. Smaller fragments migrate faster than larger fragments under an electric field. Agarose gel used to separate DNA fragments of few hundred to 20 kb in size, while polyacrylamide gel is preferred for very small fragments. Agarose gel with pulse electric field is used for very large size DNA ranging from 1000 to 2000 kb. Denaturing of DNA is performed uh, by using alkali, and this step is very essential for hybridization, because for hybridization we need single stranded DNA. Blotting will be performed then. In that, the denatured DNA fragments are transferred from gel onto a nitrocellulose or nylon membrane by capillary action. It can be done either by using electric field, which is called as electroblotting. The relative position of DNA fragments remain unchanged, and this is the benefit, and it helps in the detection also. DNA is permanently immobilized on the membrane by baking at 80 degrees Celsius in vacuum in case of nitrocellulose membrane, or by UV cross-linking in case of nylon membrane. Then pre-hybridization is performed, in which the membrane is treated with blocking solution containing phycol, polyvinyl pyrrolidone, and bovine serum albumin to prevent non-specific binding of the probe. Then hybridization is performed. Uh, the membrane is incubated with a labeled single-stranded DNA or oligonucleotide probe that is complementary to the target DNA sequences under controlled stringency condition. Hybridization stringency depend on temperature, salt concentration, and probe length. High stringency ensure binding only to perfectly complementary sequence. For high stringency, higher temperature and lower ionic strength is needed. After hybridization, the membrane is washed to remove unbound and non-specifically bound probes by using buffer of increasing stringency. Then DNA will be detected, the hybridized DNA will be detected. The membrane is exposed to X-ray film or imaging system to detect radioactive or non-radioactive signals, revealing band corresponding to target sequence DNA sequences. Radioactive probes produce dark bands on X-ray film after exposure, indicating the presence and size of complementary DNA fragments. So the membrane used are nitrocellulose membrane and nylon membrane. But here nitrocellulose membrane can only be used for the DNA. However, nylon membrane can be used for DNA as well as for RNA. Nylon membrane is more robust, and it it can reduce the baking time, as it can be cross at as DNA or RNA can be cross linked easily by UV exposure, and it is reusable. So the probes used are P32 labeled or non-radioactive probe, that is biotin, digoxygenin. So application of southern blotting are are in DNA fingerprinting, RFLP analysis, gene mapping, detection of transgene, and identification of genetic mutations. Its advantages are it is a highly specific and sensitive technique. It allows detection of single copy gene, useful for genome mapping and diagnostics. Coming to limitation, it is time consuming. It requires large amount of high quality DNA. It, u- it uses radioactive probe, which is not safe. So the related techniques of the hybridization are northern blotting, western blotting, and eastern blotting. In northern blotting, RNA is detected. In western blotting, protein is detected, while in eastern blotting, post translational modifications will be detected.